everyone, Mrs. Ortega here. I'm going to take a look at Chapter 44 today, which covers osmoregulation and excretion. The albatross that you see in that photo is able to drink salt water without any ill side effects. If humans were going to drink salt water, we would eventually experience symptoms of dehydration, eventually, if left untreated, death. So how does this albatross drink salt water without any ill effects, you may ask? And it's due to the properties of osmoregulation and excretion, which we will explore in this chapter. Osmoregulation and excretion is an balancing act. Physiological systems of animals operate in a fluid environment. They're dependent upon water for many things, one of which is osmoregulation. So osmoregulation controls solute concentrations and balances water gain and water loss. So ions such as sodium, potassium, and calcium all must be maintained at concentration levels that permit activity of normal life, such as muscles, neurons, and other body cells rely on concentrations of these ions in very distinct fashion. And so there is a given range of concentration of these ions that are acceptable for the individuals, for their muscles, their cells to be working in that environment. And so looking at different types of animals would show different types of regulations. Desert and marine animals will face desiccating environment. So marine animals being a very salty environment is going to naturally lose water, and along with desert animals in a very dry environment, their bodies can quickly be depleted of water, and so efforts must be established to maintain that water in the body. Freshwater animals have the opposite issue. They have to fight from getting too much water, and so they have to absorb salts from their surroundings and excrete large amounts of water as well. And so that brings us to the next topic here. Excretion is going to rid the body of nitrogenous metabolites or waste products. So as animals and humans take in nutrients such as nucleic acids and proteins, breaking those down, there will be components of nitrogen in the form of ammonia, or NH3, is going to be formed as a byproduct of dismantling or breaking apart large proteins and nucleic acids to rebuild for other nutrient protein enzyme needs. And so the first two things here to really understand is that osmoregulation is controlling those solute concentrations and water concentrations, whereas excretion is getting rid of waste products, nitrogenous waste. And so now we're going to take a look at osmosis and osmolarity. If you remember from Biology 1, osmosis is the movement of water across a semi-permeable membrane. And so osmolarity is the solute concentration of a solution, and this determines the movement of water across a selectively permeable membrane. And so this is measuring in terms of osmosis, the concentrations of those solutes. And so in terms of osmosis, there are different types of environments. The first one is isoosmotic environment. That's where water molecules are going to move across that membrane at equal rates in both directions. So they have the same amount of water and the same amount of solute concentrations, and so it's going to move in equal rates in both directions. If they have difference in osmolarity of two solutions, then there's going to be a net flow of water from the less concentrated hypoosmotic region to the more concentrated hyperosmotic solution. And so there's a picture on the next slide that might help you understand this process. And so here you can see a beaker with a selectively permeable membrane to water, solutes on one side, or on both sides. The left side has more solutes than the right side does. And so when you're looking at just movement of water, that's going to be looking at osmolarity. And so the side with more solutes will be the hyperosmotic side. And it's going to have more solutes and less water. Then the hypoosmotic side is going to have lower solutes and more water. And so water is going to move from where it's more concentrated to where there's less concentration of water. And so it's going to move here from the hypoosmotic side to the hyperosmotic side. And so these terms are important to understand. And so we're going to apply these terms to different environments in the next few slides. And so you want to be able to understand what these terms mean. So isoosmotic is going to be the solutions are the same. Hypo is going to be there's more water in the concentration or in a solution. And then hyper is going to be there's less water in the solution. And so in terms of animals and organisms, some are going to be osmoconformers, where they are going to be isoosmotic with their surroundings and they're not going to regulate their osmolarity content. Whereas most animals that we'll see in this chapter will be osmoregulators that will actually expend energy to control their environment in terms of hyperosmotic or hypoosmotic. In terms of their environment, they're going to be different than that of their environment, and they're going to have to spend energy to control that water uptake or the water loss in that environment. Here's an example in the picture below of sockeye salmon. They are osmoregulators that migrate between rivers, freshwater rivers, and the ocean. And so they have to migrate, they have to be able to regulate their water uptake and water loss. So we'll see in the next few slides how marine animals and freshwater animals will do that. So starting off with marine animals, most invertebrates are going to be conformers, so they're going to match their environment, but the vertebrates and some invertebrates will be regulators, meaning they're going to control their internal environment, and this internal environment is going to be static, or it's going to try to stay the same no matter what their outside environment's doing. And so the marine bony fishes are going to be an example here that they are hypoosmotic to the seawater. So they are in a hyperosmotic environment. And so these terms can be confusing, and so I like to refer to the environment. So keep that in mind. Marine animals are in a hyperosmotic environment. And so when you're in a hyperosmotic environment, that environment is going to have more solutes, less water, and so that's going to tend to move water from that organism into the environment. So they're going to tend to lose water, and to combat that, they're going to gain salt. And so they balance that water loss by drinking large amounts of seawater and eliminating the ingested salts through their gills and kidneys. And so you can see that in the image form here. Osmoregulation in the marine fish. They're going to gain water and salt from food, along from drinking water, their seawater here. And they're going to excrete some salts from their gills and some salt from their urine. But they're going to have less in terms of excretion. They're going to try to retain as much water as they can, along with that salt, to prevent too much water from leaving, as that osmotic water is going to be lost through gills and other parts of the body surface due to osmosis, because that water is wanting to move from where there's more water in the fish to where there's less water in the environment. All right, so freshwater animals will have the opposite effect. They're going to constantly take in water from their hypoosmotic environment. And so to combat that, they're going to lose as much salt as they can and maintain water balance by drinking almost no water and excreting large amounts of dilute urine, so trying to excrete a lot of that water out of their body. And so if this was not something that they're able to do, eventually the water going into the animal will eventually swell the cells too large and eventually burst those cells, so they could eventually die through cell explosion if this was not maintained. So it's very, very important. And so any salt lost by diffusion, they're going to be replaced in the food and by uptake from the gills. There's a constant intake of salt, and the freshwater fish is trying to get rid of that along with the water. So you can see that in this picture here, that they will gain water and some ions in food. They will take in salt by the gills and water through gills, now their body surfaces, but they're excreting large amounts of water and salt in their urine. And so this is because in the freshwater environment, the fish has more solute than the fresh water. And so the water is wanting to go into the fish, and it's going into the fish, and eventually could, if left untreated or unregulated, would cause cell explosion in that animal, which is would be lethal. So freshwater, there's too much solutes in the fish, and so the water is going into the fish, and the fish is trying to excrete that water back into the environment. All right, so moving on to land animals. So how do land animals regulate their osmoregulation, their osmolarity? And there are several adaptations to prevent water loss, which is key to survival on land. We've discussed that some in plants and invertebrates, their movement onto land came with many adaptations, some of which are shown here. First one being body coverings. Most terrestrial animals will have some type of body covering to prevent dehydration, like the exoskeleton made of chitin, or keratinized cells in humans provide a layer to prevent dehydration. Other animals have different methods, like desert animals will have simple anatomical features that prevent water loss, and behaviors such as a nocturnal lifestyle, being awake during the evening time when the temperature is cooler will allow for less water loss during activities than doing so during the daytime when the sun is out. They also maintain water by eating moist food, producing water metabolically through cellular respiration. In terms of cellular level, the end of cellular respiration, the mitochondria, one of the products is water. And so how do land animals regulate? They're going to be osmoregulators, and so this regulation depends on the amount of energy, which differs based on how different their osmoregularity is from its surroundings, how easily water and solutes move across the animal surface, and then lastly, the work required to pump solutes again across the membrane. So all these things contribute to why there are many different ways land animals will regulate their water balance. So the cell structures of which they will regulate this water balance is through transport epithelia. So these are epithelial cells specialized for moving solutes in specific directions. They're usually going to be arranged in tubular networks, And our example, non-human example, is through a marine bird. They have a nasal gland that will remove excess sodium chloride or NaCl from their blood. And so you can see that in this image here. So as the bird drinks in salt water, as in the albatross example in the beginning of this chapter, that water is going to be taken in absorbed through the small intestine, the colon region of their digestive system, and then that goes through the circulatory system to go throughout all parts of the body. So in the circulatory system, they'll have an increased amount of water, but also an increased amount of salt. And so as this makes its way to the nasal salt gland, you'll see there that there are capillaries in the nasal salt gland next to the transport epithelia that will remove that salt from the circulatory system. And so if you look at this closer up image here, you'll see that in the blood flow, there's more salt starting out initially than the lumen of the tube that's going to secrete that salt. And so as it moves across by diffusion, that salt's going to move out of the blood into that tubule, where then it's secreted out of that gland that's up top here. And so that's how they're able to drink this salt water without having the ill effects of dehydration that other animals would experience. But we're going to focus on how, in terms of excretion, the different metabolic wastes are excreted, and then eventually the human metabolic waste system. And so the excretory system we'll see in this chapter as well. But first, we'll take a look at the different nitrogenous wastes. They come in three main forms. First is urea, second is uric acid, and then ammonia. These all differ in toxicity levels and energy costs of making them by the organs and the tissues. So looking at urea here, most terrestrial mammals and many marine species will excrete urea. This is a less toxic version or metabolic waste than ammonia. And so invertebrates, this is made in the liver, and it's carried to the kidneys where it's then excreted. So we'll see in humans how we produce urea, and that gets excreted through the kidneys. However, this is energetically expensive in terms of molecular necessity. It's going to require some energy in the form of ATP and other molecules, and so this conversion from ammonia to urea does take a cost there. The other one here, uric acid, is shown in insects, land snails, and reptiles. Birds will excrete uric acid. It's relatively non-toxic, so that's good for the environment and other solutions, but does not dissolve readily in water. So you'll see in animals that produce uric acid, it's going to be more of a paste substance with little water loss, which is good for the animal. But it is the most energetically expensive molecule to make by the cells other than urea. And so here in this next image is a great image to look at. And it shows the metabolic waste type, which types of animals makes it, and then I've also added in terms of toxic levels and expensive levels from most to least here. And so it's also good to see here that you can look at proteins and nucleic acids break down into amino acids and nitrogenous bases. This produces the metabolic waste of the amino groups, the nitrogenous waste. That's converted to different forms by different animals. So looking at the first one here, the aquatic animals like bony fishes will convert that NH2 to ammonia, which is NH3. That's most toxic, and it's least expensive, which makes sense for aquatic animals. They're surrounded by water. This little amount that they're going to excrete will not have a significant impact on their environment. Even though it is toxic, there's going to be, in terms of relative concentration, it's going to be very, very, very small compared to the amount of water concentration that's available in the aquatic environment, and so it has no significant impact. And it's least expensive, making it the most preferred excretory product for the fish. Looking at urea here, in mammals, most amphibians, sharks, and some bony fish will excrete urea. So as you can tell by its structure, it's added a ketone group here, the C double bond to oxygen there, with 2 amino groups attached. This product is is moderately toxic and is moderately expensive. And so it's in the middle range of the groups here. And so it makes sense for the mammals and these other animals to produce it, as it's a relatively moderate aspect of both. Then lastly here, uric acid. It's the least toxic, which is good for their environment, but it's also the most expensive to produce. So these reptiles, birds, insects, and land snails have to spend a lot of energy producing it, but in some cases they do not store their waste for very long, and so that makes it worthwhile to make it the least toxic and excrete it after it's made. And so while it takes the most energetically expensive costs, it's worth it for these animals here. So overall, you do want to know these groups, these metabolic wastes, their expensiveness, their toxicity, and then which, in terms of category, which group produces it. Okay, so now we understand the osmoregulation and waste products. We're going to look at the excretory process, which comes in 4 main stages. And so most systems will produce urine by refining a filtrate that's derived from body fluids. And so the key functions of most excretory systems are shown. The first stage is filtering the body fluid. And so for our example here, you can see the filtration stage starts with a network of capillaries along inside of a filtration tube. And so that's going to filter the circulatory system, the body fluid being the circulatory system fluid there. Reabsorption will be the second portion, and this is going to reclaim valuable solutes. So in the filtration stage, usually a lot of solution is quote unquote squeezed out of whatever system it's coming from. And so you have to reclaim some of those solutes that were lost from an immediate filtration stage. And so reabsorption is reclaiming valuable solutes. Then secretion. So secretion is going to add non-essential solutes and waste to the filtrate. Note that this is not excretion, which is leaving the body. Most people, if anything, confuse these 2 stages with each other. And so taking a look now at learning at secretion is when you're moving solutes and waste to the filtrate that's eventually going to leave the body. So secretion is getting those things out of the normal body fluids, which here would be the circulatory system. Then the last stage is excretion, where that now processed filtrate can be called urine, and that's going to be released from the body. So that's getting rid of the waste. So secretion is getting rid of the solutes from the circulatory system, but excretion is getting rid of those wastes from the body. It's a big difference there. So for humans, this process is done by the kidneys. The kidneys are the excretory organs of vertebrates, and this functions in both excretion and osmoregulation. So we're going to maintain the water balance, solute concentrations, and produce waste to excrete in the kidneys. And so to do so, the tubules of the kidneys are highly organized. We'll see here in just a minute. We'll take a look at the tubule of the kidney. It's called a nephron. And so the excretory system includes ducts, structures that carry urine from the tubules out of the kidney and then to tubules that temporarily store the urine and then eventually out of the body. And so that's shown here. So we have two kidneys. These are closely attached to the circulatory system through the posterior vena cava and the aorta. And so the circulatory arteries and veins going to the kidneys are called renal arteries and veins. So from there, the kidneys produce the urine that will then be excreted. So the urine first has to leave the kidneys through the ureters, those two long tubes connecting the kidneys to the urinary bladder. The urinary bladder is where it's temporarily stored until it can be excreted out of the body through the urethra. And so looking at the kidney structure, if we were to slice one open, it kind of looked like a tomato. But this tomato is much, much different in form and structure and function, that it has these different renal areas. So the outermost area is called the renal cortex. The inner area is called the medulla. So these are going to be regions of where we'll see the tubules and nephrons will be that will filter the blood for waste. And so as it produces the urine, it's going to collect down in the center there, the renal pelvis area. And from there, out of the renal pelvis is going to go through the ureter to the urinary bladder. And so taking a look at the renal cortex and medulla up close, you can see it has many, many tiny tubular networks. These are called nephrons. So based on where they're placed in the region, if they're more in the cortex region, that's going to be called the cortical nephron. If it's more in between both regions of the medulla and cortex, it'll be called a juxtamedullary nephron. But for us, we'll look at and focus on the nephron in terms of its structure and its function. But before I move on here, do take a look. The collecting duct goes all the way down eventually to the renal pelvis, which would be down there. And so taking a look at the nephron organization, in reality, it looks like this picture here. But in terms of studying, we'll unfold this tangly network of tube and take a look at the different stages in this chapter. And so in reality, it looks like this image here, but we'll unfold it and to better understand it. But first, we'll go ahead and play a short video about the excretory system and the kidneys here.
The outer part of the kidney is called the cortex. Inside the cortex is the medulla. Urine collects in the renal pelvis and drains from the kidney through a tube called the ureter. A tiny branch of the renal artery carries blood to each nephron for processing. A tiny vein carries blood away. Blood pressure forces fluid through the walls of a cluster of capillaries called the glomerulus, and it collects in the cup-shaped Bowman's capsule. The filtrate is refined as it passes through the twisted nephron tubule. Useful substances are returned to the blood, and the remainder collects as urine. Refined urine is carried by the collecting duct into the renal pelvis for excretion from the body.
Okay, so now we're going to take a look at the nephron and how it's organized for processing this filtrate. So the filtrate is the first part that's produced. This is produced in a structure called the Bowman's capsule. So the Bowman's capsule is the tube network that surrounds the capillary structure called the glomerulus. And so from there, it's going to squeeze those fluids out of the circulatory system. And so those fluids are going to be salts, glucose, amino acid, vitamins, nitrogenous waste, and other small molecules will come out of that filtrate. And so going from the Bowman's capsule to the next structure will be the proximal tubule. And so here you can see the written description of these steps, but I'm going to zoom forward to our picture form so I can show you an illustration of what happens. And so these next slides here, right, where we were here, this one and this one also describe the process, but we'll take a look at the image. So here you can see the Bowman's capsule and the glomerulus. So that will produce a filtrate that's going to be in the tube, that's this yellow tube here. And so moving from that region, the solution that's been pulled out of the circulatory system is called a filtrate. And this is going to be regulated in the proximal tubule, which is the first tubular section the filtrate, the solution will go through. And here you can see on the side, the arrows indicate where it moves, and then the different colors indicate what type of transport it is. Active transport will require energy, usually in the form of ATP, to move a substance, whereas passive transport, those molecules can cross that membrane between the tube to the kidney structure, or to the kidney cells, into and out of the kidney cells into the kidney tube here. Also, you can see what molecules are secreted at the beginning into the tube, and so this is called a filtrate, and you can see those molecules there. And so in that proximal tubule, it's going to be reabsorbing anything that's valuable and making sure anything that's not needed or toxic is going to be staying inside that tube. And so in the proximal tubule, you can see it's passively transporting bicarbonate, HCO3-, water, and potassium out of the tube, along with actively transporting sodium chloride and other nutrients out of the tube, and then passively transporting ammonia, NH3, and hydrogen ions, H+ N. And so that's going to start the process here. From there, moving down the loop of Henle or Henle, which is shown here, the loop of Henle. So if you were to go back, it would be the descending portion happens first, then the ascending portion. And so looking at the loop of Henle here, you can see on the descending portion, the water is going to be passively transported back to the kidney cells. So this is trying to keep as much water as we can. And so the way this is going to happen is through passive transport through aquaporin proteins. So passive transport just means that energy is not required for that molecule to move across the cell membrane. And so with the help of some protein molecules called aquaporins that will move water out of the filtrate and back into the kidney cells in the medulla region here. And then as it ascends or goes up the second side of the loop of Henle, you can see here it's going to be no longer functioning for water, but for sodium chloride. So in the first thin segment of the ascending lib, it's going to be passively transporting sodium chloride to the kidney cells. as sodium balance is one of those things that is very, very important to many different types of cells, regular body cells and neuron cells. It's going to need to take in sodium chloride to an extent. And so here that is done passively without any energy input. But as you reach a thick segment of the ascending limb, it's going to have to do this actively, so spending energy to bring that sodium chloride back in. Then going to the distal tubule is going to be regulating ions and minerals for pH balance, mostly potassium, hydrogen, water can be regulated here as well. So as you can see, they're actively transporting in this section, and it's actively transporting sodium and chloride and bicarbonate out of the filtrate and potassium and hydrogen into the filtrate. And it's passively transporting water out of the filtrate as well. Then the last stage, the collecting duct. Here, the collecting duct is going to now be trying to get the last of the water and the sodium chloride out of the filtrate by process of osmoregulation. Here is also going to be bringing some urea is actually going to escape out of the filtrate and kind of remain in the inner medulla, but that will be fine for the inner medulla as it has a very, very high solution or solute concentration, and so we'll be able to maintain its structure even with urea present. And so as that filtrate leaves the collecting duct, it becomes is now called urine. From there, it will go to the renal pelvic region, where then it will leave the kidneys through the ureters. And so this process, while it may look a little simple there, can actually be a lot more in terms of information than what's actually been shown. So this is a very key trait to conserving water in the mammalian body, along with some unique facts here that this urine produced is going to be hyperosmotic to other bodily fluid. And so that's going to have more solute and less water. So that is very beneficial to land mammals as they need to conserve as much water as possible. And looking at it in terms of osmolarity, there are two primary solutes affecting it, and those are NaCl and urea. And so here to look at osmolarity in the kidney in terms of NaCl and urea, you would see quite a different picture, which is shown down here. While this is important information, this is not something I ask you to memorize, so you don't need to try to memorize this picture here. But I'd like to go over it as it might help you understand this section better. In terms of the kidneys, it has a gradient of osmolarity. So the cortex is going to be, in terms of solute concentration, it's going to have the lowest amount of solute, and then the inner medulla is going to have the highest. And so that's represented here by numbers, that the milliliters of osmolarity for liter, it starts at 300 in the lowest region and then goes up to 12,000 in the inner medulla. And so as the filtrate goes through the tubules here in the nephron, you can see there's a difference in osmolarity as it goes. As it loses water, it's gaining more osmolarity, which is matching that of its environment. As it's going ascending, it's going to be losing salt, whereas lowering osmolarity, and then at the final stage of the collecting duct, losing water, losing salt, is going to have a higher osmolarity. So it's always going to match that of its environment, which allows for this process to work. If it didn't, it wouldn't work. So It is a very, very sophisticated process that has a lot more information to it than what we just saw. And so in these slides here, that kind of goes over that information a little bit more. But this is something I don't ask you to try to understand, as it's more of more for anatomy class. But it does have a theory. It's called a countercurrent multiplier system. As this involves that loop of Henle, and that allows for it to have produces hyperosmotic fluid while maintaining as much water inside the body as we can. That urine produces isoosmotic to the interstitial fluid of the medulla kidney cells, but it's hyperosmotic to blood and other fluids elsewhere. For your reference, blood is going to be usually around the 300 range, which is the same as the cortex region there. So the kidney has some regulations of nervous and hormonal controls that will regulate the functions. And we'll look at one of those here. And it's called the antidiuretic hormone, or ADH for short. In older textbooks is called vasopressin. So this hormone is going to be very important in maintaining the blood osmolarity. And that it has there are receptor cells in the hypothalamus that will monitor the blood osmolarity, and this will regulate the release of this hormone, ADH, from the posterior pituitary gland. And so at any time when the osmolarity rises above a set point, so when water is lost, ADH will be released. So this ADH will then circulate through the circulatory system, will then go to the kidneys and will bind onto receptor molecules onto the kidney cells. This will lead to a temporary increase in the aquaporin proteins that take in water in the collecting duct cell region. And so this will bring more water back into the kidney cells, and then less water going through the urine. And so that will reduce the urine volume and lower the blood osmolarity. And so we have a picture of that here. That the ADH receptor on the collecting duct cell of the nephron is going to, when bind with ADH, trigger a cascade reaction that will then put these aquaporin proteins onto the cell surface of the lumen and of the kidney cell on the other side. And so it's going to be taking water in from the filtrate from that kidney tubule into the kidney cells. And so that's going to help bring water back into the body. And so the regulation process is shown here. So this is done through the hypothalamus. So whenever the blood osmolarity rises, such as after sweating profusely during an exercise, this hypothalamus will detect the higher level of osmoregularity. It will stimulate the thirst region in the brain to stimulate the drinking of water. That will help restore normal osmolarity in the blood, but it also will trigger the posterior pituitary gland to release the ADH hormone. ADH will then go bind to the collecting ducts and will take in more water from the filtrate that is going to make the urine. And then from there, it's going to restore the normal blood osmolarity. And so we have a short video about that here. We can go ahead and show you.
Hormones regulate water reabsorption by the nephron tubule in response to body fluid intake and loss. On a hot day, when you're sweaty and thirsty, blood solute concentration rises. In response, the brain signals the posterior pituitary to increase its output of the hormone ADH. ADH makes the walls of the collecting duct more permeable to water. Recall that the interstitial fluid surrounding the nephron tubule has a high solute concentration. ADH allows more water to leave the filtrate by osmosis and re-enter the blood. The body conserves water and concentrated urine is produced. When you have been drinking more water and not losing much fluid, the blood becomes more dilute. The brain responds by decreasing the secretion of ADH. The collecting duct becomes less permeable and less water is reabsorbed. The body produces dilute urine.
Okay. Another thing that's closely tied with this information is alcohol and how alcohol interacts with ADH. It is a diuretic as it inhibits the release of ADH. And so without the ADH hormone to bind to the receptors, water will not be taken back in by the kidney cells from the urine. And so as a result, the urine that's secreted will have the water that the body needs. And so this results in dehydration. And so with more consumption of the alcohol, the more dehydration process will happen, along with other factors like the salty snacks provided in occasion by bars, such as peanuts or pretzels, will also generate thirst. Drinking more alcohol, which will lead to more dehydration, and so that is a very detrimental cycle to the body. Another thing here is there's a mutation in ADH production that causes severe dehydration and results in diabetes insipidus. So those are just two of many things that could be looked at in terms of how the kidneys are regulated and the factors that contribute to osmoregulation and excretion. So on this next slide here shows a summary of the different types of animals, the inflow and outflow of water and solutes, and the urine production. And so if you have any questions, please let me know, but that brings us to the end of chapter 44. Everything else in the chapter, if you are looking at the textbook, we do not cover, so you can ignore those sections and stick to what we see in the slides here. But the next video we'll be posting will be chapter 49, where we look at nervous systems. So bye-bye for now.